Hello and welcome to Teach Me in 10. Today we're joined by Phil Beckett, who's a discovery imaging field application specialist with Cytiva. Phil will introduce Cytiva's image integrity checker and explain how it helps researchers, industry teams, and journal editors verify gel and blot image authenticity. He'll share some common red flags and best practices for preparing and reviewing images so you can publish with confidence.
Hello. Today's Teach Me in 10, we're going to look at how to protect your research images. So what we're going to cover today is we're going to go through concerns with the image integrity in scientific research, why it matters and how to maintain that integrity research. We're going to give you advice on safe handling of those images. We'll give you an introduction to Cytiva's free image integrity checker software. We'll look at how to improve your data submission to journals. And we'll show you an actual demonstration of how to protect the integrity of your images. So in this survey here, they looked at over 1,300 papers and found that 10% had at least one instance of image manipulation. And of those images, 80% contained a gel electrophoresis image. And that could be like a Western blot or a classic SDS page gel image. But basically, this means that over 20% of the papers that had an electrophoresis image involved some form of image manipulation. Now, it's very easy to manipulate images in something like Photoshop. So you can see in this example here that we had an SDS page gel that's transferred onto a membrane, and they're looking at a particular protein here. And in this instance, you can see they've copied and pasted one of the other bands and placed it into the blank lane just to kind of create a story to kind of hopefully back up the research that they're trying to push forward. But obviously this is fake data, so it's not believable, and it's a problem. So this has been highlighted in journals like Nature and Science, where they've looked at this problem and flagged it up as an issue and trying to address how to deal with it. There's actually big kind of think tanks on how to look at this. And then also this has made its way into the popular mainstream in things like the New York Times, where they've highlighted this as being an issue within the scientific community. And then some big cases here. You can see in this case back in 2023, the Stanford president actually resigned over manipulated research from his lab and having to retract at least 3 papers. I think there might be some more now being called into question and having to be retracted. And then there's another one highlighted here in Science, where the researcher there was falsifying, or his lab were falsifying Western blots and images. I think there was like micrographs of brain sections as well. So again, leading to retraction of published papers and getting a lot of the other data being called into question. There's a famous person here called Elizabeth Bick. So she is a science integrity consultant, and her job is to go through journals looking for images that look like they might have been manipulated or have repetition from one paper to another one. But she's like saying in a few years, AI will be able to create fake images that are undetectable for experts like me. They can take that kind of copy and paste we saw before, but then they could make that pasted band, they could make that look a lot less like the original band and make it look like a totally different band. So it's going to be very difficult to detect any differences. And Nature conducted a survey here where they asked a group of people if they thought they'd seen AI-generated images, and about a quarter of those said yes, about a third said no, but then another half were like, they were like thinking that they probably was, but they weren't 100% sure, but they definitely had that reason of doubt that something had gone on and maybe they had been manipulated. So this is obviously important, and especially now with the way that we're sharing data between different collaborators. We have cloud-based storage, but then you might edit it or go through another software package before it goes into that cloud. And then obviously you don't want to have falsified data or manipulated data, which might get called into question. So we need some way to prove that that image is, the true raw data. So like I said, accidents can happen. So it might not be intentional that you have just basically gone into something like Photoshop, you've cropped, rotated, you've adjusted the contrast, you've resaved, but then that might lead to problems in just verifying that it is the true raw data. So you really want to work with, like things like the image integrity checker to give you that confidence when you do submit the data. And like I said, here's an example where we've taken the original 16-bit TIFF image, and it's obviously got a lot of embedded information in there. You can see here in the header, there's a lot of information about how that image file was captured. But then they've gone into a non-scientific software, so something like I said, PaintShop Pro or Adobe Photoshop, whatever. And then they've gone to resave the image, and then it probably will save it as an 8-bit image. You're going to lose that metadata. And then again, it's not good for quantitation, and it's going to get queried when you submit that data into wherever they're going to be kind of looking at it, whether it's a regulatory body or a journal. So the image integrity checker tool we talked about there is a free software, and basically it's enabling you to authenticate each of the images that you have from your experiments. And basically it's building confidence in the data that you're going to be submitting to whoever's going to be looking at it. So that basically is going to help you because it's going to make it very quick and easy for them to verify that that is a raw image. You don't need any special training, don't need to be an expert in this. It's just like I said, a free software. And basically it results in a unique digital fingerprint for all your raw images. We talked about a digital fingerprint, but what we mean by that is actually a secure hash algorithm known as an SHA. These are a family of cryptographic hash functions that are published by the National Institute of Standards and Technology as a US federal information processing standard. And they're widely used in the public domain. You could be using vaccination certificates, digital currency like Bitcoin, things like that. They're also used to validate images submitted in courtrooms. And it's basically a one-click authentication. It's simple, it's fast. Like I said, you don't need any specialized training or expertise in this. It's just using the software. If you're not doing a single image, you've got a whole backlog of images you want to verify, then that's easy. You can just drop them in a folder and they'll go through it incredibly quickly. It works at any stage in the process. So like I said, you might have sent that image off to a colleague or a collaborator to look at, and they're sending it back to you. So at any point in that image's history, you'd be able to go back and look at it and check that it is still the raw data. And it's the embedded information such as the pixel values, the metadata, all remain part of the image and are used as part of that verification process. So basically it's going to generate a report that will be attached to your submission and showing that response time within the journal validating it. Yeah. So how will this improve your publication submission? So like I said, it's going to reduce the risk of rejection or revision because it's going to quickly be able to check to see if it is the true data there. It's going to build trust with the editor and reviewer. They're going to see that you're using this tool and that it is the raw data. It's going to save time because they obviously don't have to go back and forth in checking if the data's been manipulated or adjusted or changed. It's obviously a really good practice to use this approach because, like I said, they don't have to query what you're doing. You can have a separate image that you've contrasted and whatever to have for the publication, but you've always got the raw data there that they can look at and verify that it is, totally true and valid. So it's obviously giving peace of mind for everybody when you submit this data. So you can be submitting publication-ready figures with total confidence. So for the journals, the editors and reviewers who are looking at that data, basically it's going to improve the integrity of not only the submitted data, but the manuscripts that they're going to publish. So they're not going to get called into question that they're, a reputed journal for having, secure data. You're going to help reduce potential risks early on because, like I said, it'll flag up very quickly. You're building that trust between the publisher and the submitter, and it's going to reduce any post-review investigations because we know it's the true data going in from the get-go. For the industrial researchers and people in like these quality control teams, again, that's going to help with authenticating, tracing the gel and the blot images, not only within the QC department, but also the R&D department. It's also going to support a GXP workflow so that you can have that electronic digital handshake between the image capture and the image analysis. We're going to look at helping reproducibility in both the nucleic acid and protein workflows, reduces the risk in that quality control, audit, regulatory submissions. When you submit that, they're not going to call it into question because they can review and check using that tool. So it's building confidence in what you're doing with your regulatory partner. So how does the image integrity checker work? Basically, like I said, you could take a single image or you can take a folder of all those images, drop them into the tool. It's going to run through incredibly quickly. It's going to give it a pass or fail. If it's from a different imaging system, then it will just say not recognized, and it's not within the realms of this to be able to pass or fail whether anything had gone on with that image. But for the ones for the Amersham Typhoon and the Amersham Image Quant 800, you'll get to the end and you can also generate this report you see here. So it's very quick and easy to do. So like I said, the images are coming from the instruments. They'll go into the free software package, the image integrity checker, and then you have this report that's generated at the end here, which you can submit with your data that you're submitting. Yeah, it's found on the website here, so you can just click on the link and this will download to your computer. And like I said, anybody can do this. So the link is again shown here. So, and some more information about the image integrity checker. And then the two instruments I mentioned that are compatible with software are the Amersham Image Quant 800 and the Amersham Typhoon. So you have links to find out more about those instruments there. So I'd like to thank you for your time today and appreciate your time. Thank you.
Thanks for watching this episode of Teach Me in 10, brought to you by Cytiva, a trusted expert in protein research who supports scientists across Western blot and imaging workflows. To learn more about Cytiva's Image Integrity Checker, Image Quant software, and other imaging solutions for Western blots and gels, check out the links in the description. And as always, if you enjoyed this episode, don't forget to like and subscribe to stay up to date with all of our latest releases. See you next time.